Welcome to everyone. It's our third lecture, and in this lecture we will talk about movement in and out of cells. So in today's lecture we will cover the following topics. We will talk about diffusion, osmosis, and we will also discuss about active transport. So here we will take an overview of our topic. As we know that movement of substances into and out of the cells take place through cell membranes. So cell membrane act as barriers. It act as barriers to some molecules but not all the molecules. That's why cell membrane is known as semi-permeable membranes. So semi-permeable membrane or selectively permeable membrane are two same terms that we use for cell membranes. Because cell membrane allows some molecules to pass through it, but don't allow some molecules to pass. In this diagram, we can see that here are some molecules that are trying to pass through cell membrane, but cell membrane is acting as semi-permeable membrane, so it is allowing some molecules to pass through it, but it is not allowing other molecules to pass. So due to this phenomena, cell membrane maintains the equilibrium inside the cell as well as outside the cell by exchanging the matter with cell's environment according to their needs. So, movement of substances across the cells take place through cell membrane, and cell membrane perform its phenomena through diffusion, facilitated diffusion, osmosis and active transport. We will discuss each of these terms in detail. So here is a term that is known as concentration gradient. What is concentration gradient? Concentration gradient is the difference in the concentration of a substance between two areas. So, the difference in the concentration of a substance between two areas is known as concentration gradient. And it occurs when the concentration of particles is higher in one area than another area. So, the movement of particles occurs from an area of higher number of particles to an area with lower number of particles. Here is a diagram that is showing the concentration gradient. In this area we can see that here is higher concentration of particles, and in this area there are lower concentration of particles. So particles are moving from higher concentration to lower concentration, and that is the concentration gradient. Now we will come to our main topic, that is the type of transport across the cells. So there are two types of transport. One is the active transport and the other one is passive transport. What is active transport? Active transport is a type of transport which occurs when substances move from an area of lower concentration to an area of higher concentration, and for this movement energy is required that comes from respiration. As we have read in concentration gradient, that in concentration gradient movement occurs from an area of higher concentration to an area of lower concentration, but in active transport movement occurs from an area of lower concentration to an area of higher concentration. So active transport is a type of transport in which movement occurs against the concentration gradient. That's why energy is required in active transport that comes from respiration. Here are some examples of active transport. Uptake of ions from soil water through the root hair cells in plants is an example of active transport. If we quote another example, then uptake of glucose by the epithelial cells in the villi of small intestine and by the tubules of kidney in the nephron are other examples of active transport. Movement of particles by the protein molecules. So, during active transport, carrier proteins are those proteins that are present in the cell membrane of the cells. So, how the movement of particles occur by these carrier proteins? So, these carrier proteins during active transport use energy to transport molecules or ions across the membrane against the concentration gradient. Here we can see that it is inside of the cell and this area is outside of the cell. So we can see that here inside of the cell there is lower concentration of particles or molecules, and in outside of the cell there is higher concentration of molecules or particles. So particles are moving from an area of lower concentration to an area of higher concentration. So energy is required. That's why it is called active transport. Now we will talk about passive transport. So passive transport is a transport that occurs when substances move from an area of higher concentration to an area of lower concentration. Passive transport includes simple diffusion, facilitated diffusion, as well as osmosis. So these three are the subtypes of passive transport. Now we will firstly discuss about diffusion. What is diffusion? So diffusion is the movement of molecules from an area of higher concentration to the area of lower concentration. Diffusion is the movement from higher concentration to lower concentration, and it is along the concentration gradient. As we have discussed earlier that concentration gradient occurs when movement occurs from an area of higher concentration to an area of lower concentration, and same thing is happening in the case of diffusion. So diffusion occurs along concentration gradient. Diffusion is called as a principal method of movement of substances within the cell as well as across the cell membrane. So, as a result, the molecules reach a state of equilibrium where they are distributed equally throughout the area. Here are some examples of diffusion. Gas exchange in gills and lungs occurs by the process of diffusion. Movement of glucose molecules from small intestine lumen into blood capillaries of villi is another example of diffusion. Some gases like carbon dioxide, oxygen, and glucose, etc., can cross the cell membrane by the process of diffusion. So here is an important concept that a cell does not expend the energy when molecules diffuse across its membranes. So in the diffusion, movement occurs from an area of higher concentration to an area of lower concentration, and cell doesn't expand the energy. So diffusion is an example of passive transport. So in this diagram we can see that molecules or particles are moving from an area of higher concentration to an area of lower concentration. So movement is occurring from higher to lower concentration. Energy for diffusion. So as I have described earlier, that cell doesn't expend the energy for molecules when molecules diffuse across cells. So here a question comes that energy for the diffusion comes from where? So energy for the diffusion comes from Brownian motion. What is Brownian motion? As we know that in the matter, all the particles like molecules and ions move randomly all the times. As we can see that here molecules are moving randomly. This random movement is called Brownian motion. So Brownian motion is the random movement of molecules and ions. This random movement of molecules and ions produce kinetic energy, and the energy of the diffusion comes from the kinetic energy of random movement of molecules and ions. What is the importance of diffusion? Diffusion of gases is important because it makes the gas exchange possible for the process of respiration. Respiration is a process of breathing in oxygen and breathing out the carbon dioxide. Diffusion of solutes is important because it gives proper shape to plants and enables the plant to transpire. So what is transpiration? Transpiration is a process through which plant evaporates through its stomata. In animal cells, water is supplied to the cells by the process of diffusion. Water is an important solvent. All the reactions in our body occurs in the presence of water. So substances are transported across the cells after being dissolved in water. Diffusion helps to get rid of waste products, and it also allows the cells to gain the useful substances which are required for the growth and energy. What are the factors which influence the process of diffusion? Number 1 is surface area. So, larger the surface area, the greater will be the rate of diffusion. So if the surface area is larger, then the rate of diffusion will become greater. Second factor is temperature. We can see that at higher temperature, molecules will have the more kinetic energy. So this kinetic energy comes from the random movement of molecules and ions. So at great temperature, the rate of diffusion will become greater. How concentration gradient influence the rate of diffusion? Greater the concentration gradient, the greater will be the rate of diffusion. It means that if the concentration gradient will greater, then the rate of diffusion will become greater. So the fourth factor is distance. So if the distance is shorter, then molecules have to travel a short distance and greater will be the rate of diffusion. So if the distance will be shorter, the rate of diffusion will be greater. What is facilitated diffusion? Facilitated diffusion is the movement of substances from higher to lower concentration with the help of transport proteins, which are also known as carrier proteins, is called the facilitated diffusion. Many molecules do not diffuse freely across the cell membrane because of their size and charge. So such molecules are taken into or out of the cells with the help of transport proteins which are present in the cell membrane. So the rate of facilitated diffusion is higher than the rate of simple diffusion. Facilitated diffusion is also a type of passive transport because molecules are moving from an area of higher to lower concentration. So here is the difference between simple diffusion and facilitated diffusion. In simple diffusion, movement is occurring from an area of higher concentration to an area of lower concentration without any carrier proteins in the membrane. But in facilitated diffusion, movement is occurring from higher concentration to lower concentration, but this movement is occurring through the carrier proteins in the membrane. So it is the facilitated diffusion. What is osmosis? So osmosis is the movement of water molecules across a semi-permeable membrane from higher water concentration to lower water concentration. So water molecules move from dilute solution to a concentrated solution along the concentration gradient. So water molecules move from dilute solution to concentrated solution in the process of osmosis. What are the rules of osmosis? So the rules can be better understood if we know about the concept of tonicity of solutions. What is tonicity? Tonicity is a term that refers the relative concentration of solutes in the solutions. Relative concentration of solutes in the solution is tonicity. What is a hypertonic solution? A hypertonic solution is a solution that has relatively more solute and it is in concentrated form. What is hypotonic solution? A hypotonic solution is a solution that has less solute and it is in diluted form. What is isotonic solution? Isotonic solution is a solution that have equal concentrations of solutes. So in this diagram we can see that this is showing the process of osmosis. Water molecules are moving from dilute solution to a concentrated solution. Here is a concentrated solution and here is the dilute solution. A dilute solution is a hypotonic solution and a concentrated solution is a hypertonic solution. So in this area we can see that water molecules are moving from dilute to a concentrated solution. Here water molecules are shown by blue color and sugar molecules are showing by a pink color. What is the effect on animal cells by immersing them in solutions of different concentrations? So when an animal cell, such as the red blood cells, is placed in an isotonic solution, then cell volume remains constant because the rate at which water is entering the cell is equal to the rate at which water is moving out. So no water will comes in the cell and no water will move out of the cell because the cell is placed in an isotonic solution. What happens when a cell is placed in hypotonic solution? So when a cell is placed in a hypotonic solution, then water enters and cell swells and it ruptures and cell ruptures like an overfilled balloon. Similarly, when an animal cell is placed in a hypertonic solution, then it will lose water and will shrink in size. So, in hypotonic environments like in fresh water, as we know that in hypotonic environment, water enters in the cell and cell swells. So, in hypotonic environment, animals must have ways to prevent the excessive entry of water. And in hypertonic environments like the sea water, they must have ways to prevent the excessive loss of water because in hypertonic solutions, cell will lose the water and will shrink in size. Now we will discuss the effect on plant cells by immersing them in solutions of different concentrations. So water balance problems are different for plant because plants have the rigid cell walls. So most of the plants lives in hypotonic environment. We have discussed that hypotonic environment is that in which lower concentration of solute in the extracellular fluid than in cell. What is the extracellular fluids? Extracellular fluids are those fluids that are outside of the cells. As a result, water tends to move first inside the cell and then inside the vacuole. So when water enters into the vacuole, then vacuole increases in the size and it presses cytoplasm firmly against the interior of the cell wall, which expands a little. And due to the strong cell wall, plant cell doesn't rupture but it becomes rigid. So in this condition, The outward pressure on the cell wall, which is exerted by the internal water, is known as turgor pressure, and this phenomena is known as turgor. What happens when a plant cell is placed in isotonic environment? So in isotonic environment, no water will enter or leave the cell. So the net uptake of water is not enough to make the cell turgid and the cell become flaccid. Flaccid means it will be loose and not firm. What happens when a plant cell is placed in a hypertonic environment? Then the plant cell will lose the water and its cytoplasm shrinks. So the shrinking of cytoplasm is known as plasmolysis. So plasmolysis is a process which occurs when a plant cell is placed in a hypertonic environment. When a plant cell is placed in a hypertonic environment, then it loses the water and its cytoplasm shrinks. The shrinking of cytoplasm is known as plasmolysis. Here is a diagram that is showing animal cell and a plant cell. So, when an animal cell is placed in a hypotonic solution, then water enters in the cell and cell ruptured. When an animal cell is placed in a isotonic solution, then no water is entering and no water is leaving. So cell remains in normal condition. So in hypertonic solution, water is leaving the cell. So cell shrinks. Same in plants but with some differences. In hypotonic solution, water enters in the plant and it becomes turgid. In isotonic solution, no water is entering or leaving the plant, so it will become flaccid. In hypertonic solution, water is moving out of the cell, so cell become shrink, and this phenomena is known as plasmolysis because in plasmolysis the cytoplasm of the plant cell shrinks. What is the importance of osmosis for the animal cells and tissues? Movement of important materials inside and outside of the cells, like the movement of nutrients, water and other desired solutes, occurs by the process of osmosis. A release of metabolic waste products like the urea occurs through the process of osmosis. What is the importance of osmosis for the plants? So osmosis is a process that provides proper support to the plant. It provides strength to the plant and it prevents the plant cells from bursting, and it also prevents the wilting of plant. So that's all about the lecture. I hope you have understood all the topics clearly. Thank you for listening.